Hello and welcome to this session on reproducible analysis of fMRI data using HALFpipe. So my name is Lea Waller. I'm a graduate student here at Charité, and I've been working together with the awesome team here on this pipeline called HALFpipe for the past few years, and so I want to give you a brief overview of it. If you want to follow along with the live demonstration or the kind of live tutorial, I guess, then you will need a dataset for that. So I think in an earlier session, you've downloaded the Amsterdam Open Imaging cohort and the subset P of 1 of it. I am actually not sure of it. If you don't have it, maybe pause the video and just get it real quick, or any other bits dataset that might be interesting to you. So I'm going to be focusing on working with bits datasets today. The second thing that you will need is a container runtime, because HALFpipe is distributed as a container. So that means that for kind of desktop, usually the easiest thing is to just install Docker for desktop, which you can just download here at this link. And then you just have one command to run, and that will get you kind of HALFpipe, and that will get you up and running. So you should have a copy of these slides somewhere. Then I think you should just be able to kind of click the links and find out what's going on. On shared computing environments, so like your lab workstation or a high-performance computing cluster, usually Singularity is better because it is easier to configure in most cases. And in this case, you download kind of this container file, which is just like a big monolithic kind of zip of all the stuff that Docker would download. And you can either download that in the command line or with your browser. And the other thing that we need is the mask and atlas files, because HALFpipe doesn't come with those. So you have to download them separately. And usually you would do this kind of as part of your, making your research plan. So what am I going to talk about today? Before the live demonstration, I'm going to give an overview of HALFpipe, just talking about kind of the theoretical framework in which we developed HALFpipe and kind of why it sometimes makes sense to use pipelines like HALFpipe. Then I'm going just to show the example with P of 1, how to set things up. Finally, I'm going to talk about what to do with the outputs, and also what are kind of some of the use cases where you kind of need HALFpipe, and maybe also where you don't need it. So yeah, let's get started. So you are probably in this course because you are planning your neuroimaging study. You have a research question that you want to investigate, and you are currently planning how to do that, I imagine. Or maybe you already have data and you're further along. But in general, kind of research questions are something like, I want to compare different groups, relate a phenotype with brain data, look at machine learning, understanding groups that way, or maybe just to combine and compare data sets, do meta-analysis, mega-analysis, for kind of open data maybe also, or just to explore data to see kind of what kind of patterns are in there. And usually when you get started with these things, you will notice, okay, there's really lots of great tutorials out there, also lots of nice educational course videos, but it's just usually a lot of need to code and kind of write things specific to what you want to do. There's also, once you've coded it, you need to debug it, so it's kind of like really this learning curve. And the other thing that you will notice is that for each and everything you want to do to your data, to get it closer to something that you can kind of analyze, something that can answer your research questions, there's so many different options and choices for each one of those steps. So I've just put on the slide, it's maybe a bit dense, but I've just put all the different things you can do. Well, there's even more, of course, but all the things that make sense to do, or some of the things that make sense to do, I've just put on the slide. There's still more. So you would kind of start with minimally preprocessing your structural image. then also correct the functional images, do different types of denoising and scaling to improve consistency between participants also, and then also calculate different statistical maps based on your data. And when kind of you're making your own pipeline, you have to think about, okay, in which order am I doing these? Am I doing, which ones am I doing? And also which parameters am I using for each one of these? So one thing that really helps with this is fMRIPrep, which I think there was a talk before this one. And it will just take care of the minimal pre-processing for you. So this is really amazing. And so now what HALFpipe does, it will kind of do these additional steps. So spatial smoothing, Riemann scaling, confound regression, and will also calculate then afterwards these kind of statistical maps or connectivity matrices for you, and finally do group statistics. So it's kind of like putting in the things that are missing that take you from fMRIPrep to something that can actually answer your research question. And why do we need this? So it's not just that this is kind of convenient, this is like a fun tool. No, it's actually that this kind of standardization really improves science, because you've probably heard of the Botvinnik-Netzer study from 2020, where they kind of lots of different teams analyzed the same data with the same hypotheses, but no team had kind of identical workflows, identical pipelines, and no two teams had identical results. So they were all kind of different, mostly similar. And this is like a problem for science, right? We need to have consistent results. And so if everyone was using standardized pipelines, then there would be so much less differences. We would get the same results no matter kind of who sets it up, because most of the crucial choices that can affect outputs have already been made in the pipeline. So how does this look in HALFpipe? So the first step is data input and configuration. So that means kind of we wanted to streamline this as much as possible. So it's in the terminal, it's in the command line, but it will actually kind of pop up inside the command line, and you can kind of, it will kind of ask you questions about your data. So it's a bit more interactive than most command line interfaces. Then we do pre-processing using fMRIPrep. So we're basically using fMRIPrep as a software library. We're not kind of, HALFpipe does not reinvent the wheel. It's mostly using what is there to kind of accomplish the goal. And so we really thought back then when we started out already that fMRIPrep is kind of the best way to go for this. And so far it's worked really well. Then we calculate statistical maps based on the pre-processed images. But before we can kind of jump to step 5, which is group level statistics, we kind of need to look at what is our data quality like. And also what is our processing quality like, because no matter how great fMRIPrep is, sometimes there are mistakes. And so we need to, or we believe that it is important to look at every kind of data set, every participant, and kind of look at the quality. Is it good? Is it bad? We developed kind of a browser-based user interface that I'm going to show later for that. So let's jump right into working with HALFpipe, and I'm going to go to my terminal over here. I'm going to connect to my high performance cluster. To type in my passphrase. Okay. And now I'm going to request an interactive session on a node, because it's usually not a good practice to work on the head node. And so now I'm going to go to my Scratch folder, and I've actually prepared things here. So I've prepared a folder called containers, and containers has our HALFpipe latest in it. And I've also downloaded the masks and unzipped them into a folder called masks. So this is the masks.zip from the very beginning. So now let us run HALFpipe. So I'm going to type out the command. This is also in the manual and on the website for HALFpipe. But so it's generally singularity dash singularity run dash contain dash cleanenv, and these just make sure that really everything inside the container is isolated from the from your kind of local configuration, because the container is also about kind of computational reproducibility, that every time you run it you get the same results, and also no matter which computer system you run it on you get the same results. So this is kind of helping, making sure that the container stays inside the container and things are not modified. But what we do need to do is we need to pass our data into the container. So I'm going to just do, that's what my file system is called. In this case it's called fast. So I'm going to pass fast into the container. And now I'm going to go to this folder, which is this folder, and I'm going to select my HALFpipe file that I just showed, the container file. So I'm going to give it the full path to that, just in case I want to reuse this command later and I'm in a different folder. It's often good to have like these absolute paths. So as you see, I'm doing this quite fast, so I'm using tab completion. If this is a bit fast, you can always pause the video, right? So then I'm also going to tell it to use cluster, which is going to mean that it's going to generate a, it's going to run in a way that is optimized for clusters, and it means it's not going to run right away, but it's going to give me kind of a script that I can submit to my cluster queue with one chunk for each participant, which makes things a lot faster, because then when we have 200 participants, we can just use kind of 200 cores across a lot of nodes to run things all at the same time. So now I'm going to press enter, and the container is going to start. It's going to take a few seconds because it has to kind of load in all the files that are in the container and decompress them. And so now we get HALFpipe. So it's actually kind of embarrassing. This up here, it should say HALFpipe in the top, but somehow it seems broken, and I only noticed kind of today when I'm recording. So yeah, should be hopefully fixed by the time you're using it. I'm using the latest version, so as you just saw. So maybe there will be a new release by the time you are watching this video, but in general, usually the latest version is a good choice because it will have all the newest bug fixes. But once you're kind of committing to a project, it's often good to just use kind of the current release and to make sure that it works. And that you can just kind of say, yeah, we used this version of HALFpipe, and then everybody knows kind of which version you used. Okay, so now let's go again. Fast users. So you can see this is a bit different from the command line because we are getting suggestions on the bottom. And so it's actually kind of helping me go through these directories. So this is kind of like a text field, and I can type in things. And the first thing I'm being asked is the working directory, which is kind of where all my outputs are going. So in this case, kind of HALFpipe is not a bids app that will just take your bids dataset and put in some kind of derivatives, some derived images, but it's actually, we actually need a separate working directory that we can kind of have full control over. So it should also be an empty directory, or you're going to get kind of a warning message. Actually I went to the wrong folder. I need to go to scratch. I'm just going to create a new folder, so this will still work even if the folder does not exist, and I'm going to call it p_1_workdir. And I'm going to press enter. So now I'm going to be asked about my data, and so if I say no, I will be asked, okay, please tell me the path to kind of the T1 images, to the functional images. So it's going to go through each of the different file types, and then I can tell it the path, which might be like fast users scratch, and then maybe it will be like subject.ni.gz, and this will just kind of tell it that all the T1 weighted image files, it will just be everything before the kind of file extension, which is nii.gz, will be kind of the subject ID, but maybe sometimes it's also going to be sub dash, or maybe you will have a suffix, and so on. So here you can accommodate kind of any type of data and it's going to work. But any type of naming of how your data might be structured or named, you can accommodate. But if your data is in bits format, so I just undid with Control-C. So Control-C always cancels the current question and goes back a step. So I can again show this, Control-C, and I'm going back. And this is kind of useful if you make a wrong choice, you can always just go back one step. So I'm going to say yes, the data is available in bits format, and it's actually also in the same folder in my Scratch folder. Oh no, it's actually in my work folder. Just now that I think about it. So we actually have two user folders on this cluster, and here it is, DS2785. So I'm just going to press enter, and this is going to take a while. And so now what it's going to do is it's going to go through all of the different folders in kind of my bids root and find all the bids datasets and see which ones of those HALFpipe can use. And HALFpipe can use a lot of different types of files. So it's of course going to need an anatomical scan, kind of a high resolution scan of the brain for registration, because fMRIPrep needs that. It's going to need your actual functional images. And it's going to need to know which images correspond to kind of which task. So task is just a tag, so you can call them any kind of characters, any kind of character string is going to work. So sometimes task rest is also a good one to use, or rest eyes open, face matching, and so on. So we're going to see this in a second. You can also use field maps, and all the three types of field maps that are supported by fMRIPrep can be added in here. And later on, when we are doing task-based analyses, you might also want to add event files so that tell you kind of which conditions happen in your task. And for resting state things, you might need to have different masks and atlases for regions of interest. So now let's see if this is finished. Yes, so HALFpipe, so now I'm back at the terminal. HALFpipe has found 216 T1 weighted image files and a lot of bulk image files too, so 6 tasks for each participant. And so now we know it has correctly identified the bits dataset and loaded in everything. So now I get the question, do slice timing? And if I get asked, if I say yes, then it's going to look for metadata about slice timing and ask me if it is correct. But sadly for this dataset, for POP1, we don't actually have any information about the slice timing. So this was is not in the bits dataset in the JSON files. It's sadly also not in the paper. So we actually have to say no and skip this step for now. Maybe at some point we'll find out. Now I get asked the next question. This is going to go on for a few questions. So here's, do we want to remove initial volumes? And so we know for this data set, we don't have to do that. That was already done automatically by the scanner. So I'm just going to confirm the 0. But I could also, for example, if I wouldn't know, I could ask fMRIPrep to detect how many kind of non-steady state volumes there are at the beginning of the scan and remove those with some code. But I'm going to go with 0, so I'm just going to press enter, and I'm actually, as you can see, this is the arrow key, so I can just navigate left and right, and I'm going to press enter. Now it tells me it found no field map image files, so because this dataset does not have field maps, which actually makes things, actually isn't that bad for data quality. Usually you're fine without field maps, but sometimes it can really help make those regressions really perfect, so sometimes it's good to have them. And now we get asked the question, specify first level features. So if I could press yes, I will be asked about the feature type, because HALFpipe can calculate a lot of different types of statistical maps of the brain, and also can calculate connectivity matrices. Wow. So then we have to kind of ask ourselves, which ones do we want to run? So so before, of course, we do any of these, before we calculate any of these statistical maps, we HALFpipe, of course, runs fMRIPrep, which, as has been discussed in a previous talk today, does all of these steps to your data. So it's actually really cool that it kind of skull strips and normalizes your T1 image. It estimates all the different kinds of spatial motion, distortion, and alignment for your bold image, and then does resampling in a single step, which is, I think, really great for rounding error, to reduce that and also to reduce kind of computational burden. And it also generates lots of really great confound time series that we can use for denoising later on. But as I also showed you before, there's some extra pre-processing steps that make sense to do, and HALFpipe gives you some suggested values for what to do. So for example, we recommend doing spatial smoothing at 6 millimeters, doing grand mean scaling, which helps kind of make sure that the range of values is comparable between subjects, that you can kind of like put everyone together in the GLM later on and values are comparable. ICA denoising, we recommend to do it, but sometimes it can be good to turn off, which HALFpipe allows you. So these are just suggested values, you can always configure them. And temporal filtering, we actually have different defaults depending on which type of statistical map you are calculating. And finally, confound regression is disabled by default, but it's fully supported. And so what is really, I think, important about these additional or extended pre-processing steps on top of fMRIPrep is that we really take care to not kind of reintroduce artifacts. So the pipeline, so we wrote HALFpipe with kind of a lot of care about the results, that kind of running multiple steps of denoising or pre-processing, like, because it's a modular kind of way, right? The next step doesn't know about the previous step oftentimes. So actually, it was shown a few years ago that this can reintroduce artifacts. So kind of when you do temporal filtering here, it might reintroduce some noise that you already removed with ICA aroma, and so on. But HALFpipe takes care that this does not happen. So HALFpipe has been written with a lot of care to kind of make sure that our denoising is, works as it should. But it's a really interesting paper. I can really recommend to check it out. It's also super interesting about how kind of, the secret life of the data underneath what you see, the commands that you run. So the other thing that HALFpipe allows you to do is kind of a streamlined, efficient multiverse analysis. So what is multiverse analysis? Multiverse analysis is kind of comparing different pipelines and seeing what they do to your data. So for example, you could, for Enigma task-based fMRI, which is kind of the project I'm working in, we are comparing 3 different pipelines, one with ICA aroma, and 2 that don't have ICA aroma, but instead use either motion parameters as a substitute. So we add them into our task-based design matrix, or we do kind of denoising and we just don't do anything in terms of denoising or confound regression. And this really tells us, okay, what is the specific impact of doing that on our results? So it actually really helps us to make sure that what we are doing is robust to these choices. Am I doing ICA aroma? Am I not doing ICA aroma? This will kind of help us understand. I'm sorry, I have a bit of allergy. So this will really help us understand what are these steps actually doing to our data, and not just in general, but in the specific case of what we are doing. So this is supported by HALFpipe, but not enabled by default. So let me go back and just show you setting this up for an example task-based model. So task-based means that we have kind of a task in the scanner where people have like different cognitive tests or cognitive conditions that we are comparing. And I'm just going to call this one. So each feature needs a name. So it's kind of like an ID. And you can just make this up. It could just be a running number. So we could call this feature number 1 and then just increment it. but usually it's kind of good to give these kind of, I'm going to go back with Control C. I'm going to give it a descriptive name, which is going to be faces task or something, or maybe just faces. And I'm going to now, with the space bar and the arrow keys, deselect all tasks that are not my faces task. So as you've heard before, PUP comes with kind of lots of different tasks, and I'm just going to, for this example, use the faces task. And now that I've selected all of these, space deselects and reselects again. And this is also shown down here, right? It tells you kind of space will toggle these kind of checkboxes, and then you can use the arrow keys. And now I will press enter to confirm. And next it will ask me about kind of the different conditions, kind of, in this case, the different types of faces that were shown to participant that we want to add to the model. And those are actually these 5 different emotions. We're just going to press enter to confirm because those look correct to me. And now I'm going to be asked about contrasts. So this is kind of which conditions do I want to compare. And I actually just want to compare faces against baseline. So I'm going to call my contrast. It needs a name, just like the features need a name. Everything kind of needs an ID in HALFpipe, and I'm just going to compare them against baseline, which is this contrast where I just put in 1 for every value. And you can also, again, with the arrow keys navigate, and these are like text boxes, and in blue you have your cursor. And once you're finished, just enter to confirm, and we do not want to add another contrast. And now we are asked kind of if we want to filter our design matrix. Let me just check if the recording is still going well. Looks good. For kind of these types of block designs, like the faces task, going with this default is actually quite okay. So I'm just going to confirm this. But sometimes you might want to do different kind of values. And now I get asked about kind of the types of pre-processing settings for the additional extended kind of steps that I just showed, what we want to do. So do we want to apply smoothing? And the default is just yes and 6 millimeters. So I can just press enter to confirm this. I could also enter a different number here, like 8 or 12 or 0 to disable, then I can also select no. So then grand mean scaling is just preset for task-based data. We think this is just the best setting, and we also think that you should use a Gaussian weighted type of filter. So we also implement frequency based filters, which are a bit more kind of strict in terms of what they include. But for the task models, we kind of just generally recommend using this filter because it's also the filter that is used by FSL to kind of filter the design matrix. So then I'm just going to keep this exactly the same for both the design matrix and our imaging data down here. So I'm just going to confirm these settings that we are going again with a 125 second high pass filter. And for confounds, I'm also going to go with the default. So I could, again, with my spacebar, kind of select different ones, add motion parameters to the design matrix, disable IC aroma, and so on. But I'm just going to confirm this. And now that I'm finished, now HALFpipe knows it's going to generate this kind of faces contrast for me. It's going to generate the task model for me and the statistical maps at the end. And now I can add another feature. So there's seed-based connectivity, dual regression, and so on. So, so what actually do we have? So we have task-based activation, which is what is the difference between conditions is our research question, right? We have different task conditions like angry faces and happy faces, or maybe also which voxels respond more to faces than geometric shapes. And then we need event files, so BIDS usually will provide them for you in a format that HALFpipe can automatically detect, but sometimes you might need to load them in manually. So in that case, you would have either just a file that contains all of them, or one file for faces, one file for shapes. And HALFpipe supports a lot of different formats for how you might kind of enter these event files. You also need to have the high pass filter cutoff. You remember we just went with the default. And in general, the rule of thumb is 1.5 times how long the slowest condition is, kind of the slowest repetition in your task. And that makes sure that the filter doesn't remove anything of your task, but only kind of the slow kind of trend in the signal. So then, contrasts of interest, you also need to know which conditions to compare. Just now we did faces against baseline. For a different task, you might do faces against shapes. And as outputs, you get GLM outputs. So the GLM outputs, this is actually kind of a bit of a dense slide, right? But you get a lot of different outputs. So you will get your design matrix and your contrast matrix. And that will kind of tell you, okay, for each volume, what kind of regressors do we have? And you can also like load these into any program and inspect these. So this is also really good for just like seeing what was actually done with your data. So next, the GLM, after it's being run, you will get effect and variance maps. So these are actually what you would pass to group statistics. So because the effect is kind of the estimated statistical map, and the variance is kind of the sampling variance, the error of the estimate. So the idea is kind of that if we would repeat the experiment, this is how much we assume that the estimate would vary, and this really helps us with doing hypothesis tests later. So basically, because based on the effect and the variance, we can calculate a t-value by dividing them, or dividing the effect by the square root of the variance, and that gets us kind of a student t-distributed t-value. And we can use that together with the degrees of freedom to do a hypothesis test. And HALFpipe, or well, in this case, FSL also calculates this hypothesis test for you, but it doesn't actually give you p-values, it gives you z-values, because those are scaled in a much nicer way and also much better to kind of store, because you don't have so many zeros before the comma that can sometimes make the precision hard to store for the computer, so z-values are much better to store. And then those are kind of normally distributed and much easier to work with. Also for kind of cluster correction or other types of multiple comparison correction that you want to do, so that kind of for all the different voxels that you are running your GLM in, right? So those are the things that you will find in your output folder or in the derivatives folder of HALFpipe. Other things you can calculate for resting state, for example. So these are in essence kind of also based on the GLM, but in this case for resting state data. So instead of comparing conditions, we are looking at which voxels show a correlated signal with a region of interest in the brain. So we also call this the seed region. And so you need a mask that kind of tells HALFpipe which voxels correspond to that region. Or you can also look at which voxels are associated with a specific brain network in an individual. But then you need network templates kind of for that. And for both of these, you will get GLM outputs. You can also look at atlas-based connectivity, and this will kind of tell you which pairs of brain regions have correlated signals based on the brain atlas that kind of gives you all the different regions. And what we get out of this is we will get kind of a file with all the time series for all the atlas regions. So these are just like the averages across all the voxels in that region. And you will get a covariance and a correlation matrix. So the correlation matrix is what you want to use when you want to compare participants, because it's standardized. But for some techniques like partial correlations and different ways to estimate the partial correlations, you actually need the raw covariance matrix. So we also provide that. Finally, what you can calculate with HALFpipe is also regional homogeneity. So kind of research question, which voxels have signals that are correlated to their neighboring voxels. Also the fractional amplitude of low frequency fluctuations. So what is kind of the contribution or proportion of those low frequency fluctuations in the total signals. And for those you get kind of also different maps out. So yeah, before we jump to the quality assessment, I might just show you really quick how to actually set these up. So I'm jumping back to my terminal, and so I'm just going to show a seed-based connectivity. I'm going to give this feature a name, which is, let's just go with the default, which was seed core. So you always get something suggested. And I'm actually only going to do this for my resting state data. So as before, using my spacebar and my arrow keys to kind of deselect everything except the resting state scan. I'll press enter. And now I'm going to navigate again to my last folder, which is Scratch, users, my username. And one thing I haven't shown yet is that when you kind of get multiple suggestions down here, you can also use your arrow key, like to jump down from the text entry down here. You can just navigate to a suggestion and press enter to select it. And now we can do this again for the masks folder. And now you see there's lots of different seed masks. So actually I'm just going to tell it to use all of them. So this is the same thing I kind of teased earlier with the what's it called? With using datasets that are not in bits format. And so I could just select one seed, right? I could just navigate with my arrow keys and just select one file in that folder. So there's kind of maybe, let's choose the left amygdala. And then that's the name of the file. But I can also use kind of this seed wildcard, and that's just going to give me all of the seeds. And so I'm going to tell it everything before the _seed _2009 and then file extension is the name of the seed. So then I actually get all 55 seeds, and I'm going to get 55 kind of GLM outputs for each participant. So then I'm just like safe. I have seed connectivity for all the regions that are kind of interesting for what I'm studying. I'm going to press enter. And it's also going to now check that my masks are in the correct space, and it's also why I have the 2009 in the name, because those are in MNI 2009 space, which is sometimes important. If it's not in 2009 space, it will automatically be converted to 2009 space. So that's why it asks you, because yeah. So now it also will show me that, hey, I loaded in all of these. And I can also, like, again, use my, maybe actually I'm not actually interested in the, I don't know, in the right medial orbital frontal cortex anymore. So I'm just going to disable that one. And while I'm at it, I'm also going to disable the left one if I can find it. Here, that's the left one. Let's also disable that one. I'm not interested in that region anymore. And I'm going to press enter. And now I've confirmed it. I get asked now about kind of the minimum coverage. So because each participant has a different brain mask. So when we do processing, we actually kind of don't know in lots of cases that every participant has the same coverage of the brain, because there's people that have bigger heads, people that have smaller heads. Sometimes people don't fit in the kind of field of view that is pre-configured for the scanner, because often, like when you start the study, it's kind of hard to anticipate how big are the heads going to be of everyone that appears, right? Sometimes also there's user error in terms of kind of where you put the field of view, so sometimes parts of the head can be cut off. So especially in terms of like regions that are really at the edges of the brain, it can be really useful to just like check, hey, do we even have data for this region? So I'm saying here, we want to have 80% coverage, so 0.8 for all of the brain masks, for all of the seed regions of interest. And otherwise, we're going to skip doing that seed connectivity for that participant. So this is really useful. And now I just get the same question about pre-processing settings. And I'm going to, again, go with the defaults, but I could, again, change these. So 6mm smoothing. I'm going to apply a temporal filter. And now in this case, I actually get more choice. So I can actually also choose the frequency-based one. So we disable that for task-based data because we think it's kind of good to kind of nudge people to make good choices. But I'm going to again go with the Gaussian weighted filter and the default of 125 seconds, just to be consistent between my resting state and my task analysis. I'm also going to use IC-Aroma. Press enter. And now I'm finished setting up, and I will get all my task contrast will be calculated, my resting state contrast will be, my resting state seed connectivity maps will be calculated. And now I could also do this for dual regression, atlas-based connectivity, and I can also set up maybe some regional homogeneity. I'm also going to go with the default feature name. And again, I'm only going to do this for resting state. And now it's like actually much simpler because I just get asked straight away about the pre-processing settings. So there's actually not much that we need to calculate reho, so we don't need to give it any files or anything. Here we actually also give you some suggestions. We actually force you to use sensible frequency cutoffs for the temporal filter, just so that this makes sense. I'm going to press enter. And now I think I'm finished with the specifying the features that I want to get out. I'm going to press no. I don't want to add another feature. And now I can also do the same thing. So if I say yes, please output preprocessed images, I will be asked again about the settings, smoothing, scaling, temporal filtering, confound removal. And so then I can also configure that. Maybe if I want to do analyses in a different software, then I can just tell it, hey, please output these images for me. But I'm just going to say no for this. And now we're already at group level model. But before we can get to the group level model, we actually need to do interactive quality assessment in the web browser. So I'm going to show this now. So here we have our web browser, and I've just navigated to the reports folder. So in your working directory, you will have a folder called reports. And in that, there will be an index.html file. And you can just open this in any recent web browser, and we'll show you kind of this overview of your data. We'll show you that kind of processing has succeeded for the subjects. And you might also see if there have been errors. It will also kind of show you an overview of the different kind of QC images that we have that we ask you to rate. And right now, all of the images have no rating. And then there's good, uncertain, and bad, which we haven't rated any images. And then down here, we can also see kind of the image quality metrics. And actually, if I go on this with my mouse, I can also hover over the dots, and I can, for example, see kind of which participant has this really high motion here. So this one is, for example, the working memory task, which I guess just now I did not enable. So this is actually from a previous run where it did calculate things based on the working memory task. But interestingly, people moved a lot during the working memory task, right? There are some participants that just moved a lot during that task, which is interesting. So we can see this on here. We can also like click them. I'm not going to do that right now, but then it will take us to exactly the images of that participant. Instead, I'm going to use this menu on the side. I'm just going to go to Explore Reports, which is just going to show me kind of this kind of preview of all the different images for each participant. And for each scan, we get kind of 6 different images. And we'll want to rate them as either good or bad. And then there's also the uncertain option, which is kind of the equal phase, which is kind of still going to include your images, but means that you will have them marked in a different way so that you can later on show them to a colleague or so. So I'm just going to go back to the charts view now. And now we see that we have actually marked all of the different task scans, so the working memory task, the resting state task, and also the phases task of that participant is uncertain because the skull stripping, we marked that as uncertain. We can also mark that as good because actually I think it looks really good. And if I want to kind of look a bit closer, I can also click to zoom. And that will just like open the picture in full screen, and I can also like move along with my mouse. And so, but this is still lots of clicking. So actually to do this really fast, I can work on my keyboard, and I can use D to jump to the next image. And then I can use W to give it a good rating, S to give it uncertain, and X to give it a bad rating. And I can also use A to go back. So it's like W, A, S, D, and X are the keys that kind of work with this interface. And this is also all documented in the quality control manual. There's also examples for good images and bad images, what to exclude, what to include, kind of what are our recommendations for those. And for this one, for example, you would just look at, does the brain atlas match the registered image of that individual? And then you will also look at the TSNR of the data. You will look at the carpet plot. So this is kind of all the different voxels plotted by brain region. You will look at ICA-based artifact removal, and also at the spatial normalization of the functional image. So yeah, and once you have finished rating all of these images, you can just go here and click export. And that will just give you a file that it's going to download, and that's just going to be called exclude.json. Well, and apparently I already had some on my computer, so I just added a suffix. And then this JSON file, actually let's not do that. will then just contain all the ratings, and you can just put that back in your working directory, and then when we do group analysis, it will be automatically used to inform kind of which subjects to include. So I would then basically on my laptop, I would download the exclude.json file generated from the web page, which is all contained right in the reports folder. It's nothing on the internet, it's all on your local computer. And I would then transfer this exclude.json file to my high performance cluster here, to my node, and put it in my working directory. And HALFpipe will automatically recognize it and use it when running the group model. So now I'm just going to specify a really quick group model. And there's actually two modes. So one is linear model, and you can kind of just like load in a spreadsheet with covariates and so on, and also do like a flexible factorial design with interactions and contrast between groups and so on. But you can also run the intercept only model, which is just really simple, and it will just calculate the mean of the group based on a mixed effects regression. And again, we can not just exclude participants based on our ratings and the quality assessment web app, but you can also kind of do this based on image quality metrics, in this case motion. And so then I'm going to be asked about kind of what are the cutoffs for motion and so on. And when I just confirm the defaults, I can also edit these, of course. Then I'm finished. And if I now press no, then HALFpipe will exit. It will tell me lots of warning messages because it doesn't know about the physiological image, the physiological files that are part of the P of 1 dataset, because we don't support that yet. And now it's going to generate a cluster submission script that I can submit to my queue, and that will run to generate all that data for me, generate all those statistical maps that we configured for me, and to do the group analysis. Okay, so that was the interactive part. And so then kind of you saw you can generate pre-processed images, and those are useful if you have kind of your own feature extraction, your own statistical map calculation scripts or software that you need or want to use. You could also just not run group statistics in HALFpipe and just look at the raw statistical maps for each participant, which like the task contrast for each participant, and then run your own group analysis with a software of your choice. And you can also share these or put them together from different data sets to do a mega analysis. And then for group statistics, you could choose to just simply report the results. So that means you would threshold and visualize the statistical maps and extract kind of where are your clusters and where are your peaks. But you could also share those group statistics with others for meta-analysis, which is also a really great use case for HALFpipe because it really makes sure that everyone is doing the same kind of processing. And this is actually what we developed HALFpipe for. So then, when do you use HALFpipe? So HALFpipe is really great because it does this automated generation of processing workflows, which gets you all these benefits that kind of you have reproducibility, and it's objective, it doesn't depend on who is running it. But it also means that actually we can only generate what we know about. So, for example, non-standard data, like the physiological files you just saw, maybe multi-echo data, might not be supported yet. It also means you don't need bits conversion. You can just enter kind of your data in the user interface by responding to the different questions and so on. And it will run fMRIPrep anyway, even if your data is not in bits format. So this is maybe useful. But it's not really like a full bits converter. So it's not just going to organize your data for you and fix everything, right? It's really focused on getting you those statistical maps. That's kind of the goal. So we have suggested values for which pre-processing steps to do and their parameters, but it's still completely user adjustable. So even if you can't adjust it in the user interface, because we kind of fix it there, just also to make the user interface a bit less overwhelming and complex, you can always kind of still do that in the configuration files by editing those. It's actually called spec.json, the file, just in your working directory. It's also, I think, documented in the README on the GitHub page, which I'm going to show in a second. So yeah, HALFpipe supports standard statistical analyses for both resting state and task-based data, but non-standard analyses, like, for example, psychophysiological interaction or dynamic causal modeling, you would still need to do that kind of yourself. So it doesn't really like do that for you, right? So if you kind of have non-standard plans, HALFpipe can still get you kind of close to that, but it can't do that. We also have the interactive quality assessment in the web browser, and that, I think it really makes it quick to look at reports, right? You can just jump through the images, look at each one, and there's not much overhead. You're basically spending most of your time looking at the images and rating them. But it also means you really need to manually look at your data. So it's going to take about a minute per participant, and that can get annoying for big data sets. But I think it's not actually a con. I mean, it might be annoying. It's actually really good to look at your data, right? It's actually something really important. So we have a detailed manual that explains every step. Actually, the link is on the next page. But of course, that manual may not yet cover your specific use case. Another pro is that HALFpipe is open source and also supported by kind of multiple Enigma Consortium working groups. But it also means that there's no kind of dedicated development and support staff. So it's mostly just volunteers who are also using this for their projects, and of course me, who is kind of leading development. So just to manage expectations. Yeah, so technical requirements, you saw it can run in parallel on the high performance cluster on multiple nodes and cores. It will take 4 to 6 hours per subject, but that depends on the data size and resolution. It could be faster, it could be slower. It will need about 8 gigabytes of RAM, but if you have kind of larger data, you might need more. And storage, I think it's about 1 gigabyte of storage for output files per subject, but of course you can also just like only output the statistical maps, it will be less. But you could also, if you have lots of storage or a small data set, optionally also store intermediate files, and that will get you kind of, will use a bit more storage. So, okay, so here are the links that I promised. So you can go to the HALFpipe GitHub page. There we have kind of the README file. If you scroll down, it will contain kind of all the first steps for getting started, what I just showed, how to download the container. There's also the manual, which is kind of a lot more detailed and will really take you through every question that is asked in the user interface and give you some recommendations for what to do. There's the task-based manual. There's actually also a resting state manual that I haven't linked here. And there's also the QC manual, but I think those are linked in the task-based fMRI manual, so if you scroll down a bit. And finally, there's the paper, where we really also talk about what I just talked about in much more depth. And yeah, you can find that here. So, of course, this was all a team effort, so I really want to thank everyone who helped with this, not just the people who helped with kind of development and planning this project, but also all the beta testers who kind of tried many iterations of HALFpipe until it worked. And yeah, if you have any questions, please contact us at enigma@charité.de. Yeah, thank you. Okay, that's it.